The overall goal of the Capture Compound Mass Spectrometry experiment is to capture and identify cyclic di-GMP binding proteins. This is achieved by fractionating a cell culture by French press and removing the free cyclic di-GMP using a PD10 column. As a second step, the capture compound is added and activated by UV light to covalently crosslink the cyclic di-GMP binding proteins bound to the capture compound in either the absence or presence of an excess of free cyclic di-GMP. Next, the capture compound is bound to streptavidin-coated magnetic beads in order to intensively wash the captured proteins. Finally, the captured proteins are digested and prepared for mass spectrometry to identify the generated peptides. Results are then analyzed in silico.
The main advantage of CCMS over existing methods like co-immunoprecipitation is that the capture compound improves the specific capture of direct partners and allows harsh washing conditions thanks to the covalent cross-linking.
Though this method can provide insights into bacterial cyclic di-GMP signaling, it also can be applied to other systems such as nucleotide signaling in general and virtually any molecule fused to the capture compound.
Prior to starting this procedure, prepare and lyse P. aeruginosa cells through a French pressure cell. Ultracentrifuge the cell lysate at 100,000 times G for 1 hour at 4 degrees Celsius.